Development of Inflamed Endothelium Targeting Nanoparticles to Treat Peripheral Artery Disease by Lana Abdulhadi.
And Sebastian Kim.
PAD is a progressive vascular disorder characterized by atherosclerosis in lower extremities, with greater than 200 million people affected worldwide, 80 million in the United States alone. This research proposes a non-invasive drug delivery system for delivering therapeutic agents to targeted ischemic muscles for PAD. This multi-phased approach holds promise not only for PAD, but addresses a range of inflammatory diseases, cancer treatment, and wound healing. The experimental test plan consists of three main studies: fabrication and characterization of nanoparticles, cellular uptake in vitro, and biodistribution in vivo. Stage 1: PLGA nanoparticles are synthesized using a standard double emulsion technique, followed by conjugation to a targeting moiety through covalent coupling. The supernatant is preserved for assessment to compare its efficiency with that of uniform PLGA nanoparticles. Results indicate an increase from 190 to 205 nanometers after characterization, suggesting successful binding, along with differences in charge from -28 to -24 millivolumes, indicating chemical interaction between the components. Additionally, the decrease in the PDI value from 0.162 to 0.161 suggests that both the nanoparticles are homogeneous in size. Stage 2: in vitro simulation starts by activating cells after incubating them in TNF-alpha for 24 hours to mimic PAD conditions, then exposed to nanoparticles loaded with green dye. Figure 1 displays the results of time-dependent study under static conditions, illustrating the accumulation of nanoparticles imaged at 20x magnification with fluorescent microscope. Visually, after 10 minutes of exposure, there's a higher presence of targeting nanoparticles compared to non-targeting nanoparticles. At 30 and 60 minutes, the nanoparticles became saturated for both the groups, show similar signal of the image. After conducting t-tests, the lower the p-value, the higher the statistical significance and certainty of the observed differences between the two groups. Our p-value was less than 0.001, indicating significance and stating that targeting nanoparticles uptake was much greater than the non-targeting. Figure 2 illustrates the results of a time-dependent study under the flow conditions, aimed at simulating the path of blood through the circular system that our nanoparticles will encounter in vivo. To achieve this, activated cells seated in 6 well-Ibidi channels were subjected to nanoparticle solutions with 2 dyne per centimeter square of shear stress. This image shows the extent of binding and targeting nanoparticles. We analyzed those images for targeting nanoparticles, showing 30% greater mean fluorescent intensity, indicating a 30% higher nanoparticle uptake in our targeting nanoparticle group compared to the non-targeting.
So next we're going to talk about stage 3. After in vitro testing, we moved on to PAD mice model by inducing ischemic injury by ligating the left femoral arteries and branches, leaving the right hind limb untreated as control. Nanoparticles were then administered intravenously through the tail vein. After 1 hour, an in vivo image was taken as shown in figure 3.a. There is more accumulation in the pad limb as shown in the red circle with the targeting nanoparticles indicated by the red intensity. After that, we collected the gastrocnemius muscles and did an ex vivo image. Some of the nanoparticles also accumulated in the liver, which is expected since we did IV injection. After that, we homogenized, lysed, and studied the nanoparticle uptake using fluorescence spectrometer. When we inject the drug containing the nanoparticles, it circulates the body through the bloodstream, but targeting nanoparticles are able to recognize and bind to specific targeting sites faster. You can also see a similar pattern present in ex vivo study shown in figure 3.b. You can see that there is an increase of about 60% in fluorescent intensity in the ex vivo image and 25% in fluorescent intensity in homogenation. Moving on to the next figure, figure 4 just again shows how effective targeting nanoparticles are as compared to non-targeting nanoparticles. Using a fluorescent microscope, an image was taken in a section muscle tissue that was sliced in thickness of 6 micron. Then an area of nanoparticles were measured and quantified, as you can see on the graphs indicated in orange color for nanoparticles. The fluorescent intensity is higher for targeting nanoparticles, so this indicates that there are more nanoparticles present. Compared to the non-targeting nanoparticles, targeting nanoparticles displayed a closer accumulation of nanoparticles in the muscle tissue. In conclusion, our research has shown that our drug delivery system effectively targets activated cells in PAD mouse models both in vitro and in vivo. Further in vitro studies will evaluate the compatibilities of our targeting nanoparticles, including assessments of cell viability and hemocompatibility. In vivo, toxicity will be examined through monitoring of parameters such as body weight, hematocrit, toxicity biomarkers like troponin, serum creatine, and lactate dehydrogenase in various organs including the heart, kidney, and liver.